Imagine if curing cancer didn't actually require months of agonizing million-dollar lab work.
Right, which is, the dream.
Exactly. What if a single injection could just turn your own bloodstream into, like, the most advanced biological manufacturing plant on Earth?
It's a staggering concept. I mean, we're talking about completely bypassing the traditional laboratory here.
Right. Welcome to the deep dive. Today our mission is to explore what is truly a mind-bending leap in medicine. We're looking at a stack of sources that ask this really radical question. Like, what if we could reprogram your immune system to fight cancer right inside your own body with just one single shot?
Yeah, instead of extracting cells from a patient to engineer them outside, we're basically treating the patient's body as the laboratory itself.
Which is wild. And to understand how scientists are actually pulling this off, we've got a really fascinating stack of sources to guide us today.
We do.
The main event is this massive new breakthrough paper detailing the mechanics of, it's called in vivo site-specific engineering to reprogram T cells.
Right, in vivo meaning inside the body.
Exactly. And we're also layering in some critical research notes on something called Anktiva, which is this really powerful immune-boosting agent. Plus, some super cool data on codon optimization algorithms for improving these CAR designs.
It's a lot, but it all connects beautifully.
It really does. Because we aren't just talking about a new pill you swallow here. We are talking about writing a software update for your cells while they are still circulating in your veins.
Yeah.
So, okay, let's unpack this.
Yeah, to really grasp the magnitude of this in vivo approach, we kind of have to start with the current state of the art.
The baseline, right?
Right. So most people following biotechnology know that CAR T-cell therapy is already this incredible miracle for certain blood cancers.
Oh, absolutely.
But, and this is a big but, it has a massive glaring bottleneck.
Yeah, the logistics are just a total nightmare. I mean, first, you have to physically extract the patient's T cells.
Right, which takes time.
Then shift them to a specialized facility, genetically engineer them to grow this new cancer-hunting radar, the chimeric antigen receptor, or CIR, and then you have to multiply them by the millions in a lab before finally shipping them back to the patient.
And beyond the sheer cost of all that, and the weeks of waiting, which some patients literally don't have, there is a fundamental biological flaw with that whole ex vivo process.
The out-of-body process.
Exactly. When you take a patient's T cells out of their natural environment and you force them to multiply rapidly in this sort of artificial lab dish, you cause immense stress to those cells.
Yeah, I was trying to visualize this while reading the sources, and it feels a bit like pulling soldiers off the front line, shipping them across the world to this grueling, intense boot camp where they're just forced to run drills 24/7.
That is a perfect way to look at it.
And then dropping them back into the war zone. By the time they arrive back in the patient's body, they're already exhausted.
That is exactly the dynamic. In cellular biology, we actually call that exhaustion differentiation.
Differentiation, okay.
Yeah, the lab expansion pushes these T cells way too far down their developmental pathway before they ever even see a tumor.
So they're basically aged prematurely.
Basically, yeah. And because they are so differentiated, they lack long-term persistence. I mean, they might fight really fiercely for a few weeks, but they burn out.
And when the engineered cells burn out, the cancer can just return.
Exactly. What's fascinating here is that this exhaustion is a major reason why these incredible therapies sometimes fail or patients relapse.
Which, raises the really obvious question. If the artificial boot camp is what exhausts the cells, why not just train them while they're still on the battlefield? Like, why hasn't doing this inside the body worked yet?
Well, because the moment you inject genetic engineering tools directly into the human bloodstream, you lose all that absolute control you had in the lab dish.
Right, the body is a messy place.
Extremely messy. You face two immense hurdles, basically, delivery and precision. And historically, we just haven't been able to master either of them safely.
So let's talk about the failures then, because the sources detail a few previous attempts that just really didn't cut it.
Right.
Lipid nanoparticles, or LNPs, are one. We know them from the recent vaccines. They're essentially just tiny fat bubbles carrying genetic instructions.
Right, and LNPs are fantastic for certain applications, like vaccines, but for CAR T therapy, they generally only provide transient expression.
Like a temporary tattoo.
Precisely. The LNP delivers the instructions, and the T cell expresses the cancer-fighting radar for a little while, but the DNA isn't permanently woven into the cell's master genome.
Oh, I see. So as the cell divides over time...
The instructions dilute and fade away, the radar just disappears, and then the cancer comes back.
Okay, so if you need permanent ink, so to speak, you have to look at viral vectors, because viruses are literally designed by nature to permanently insert their DNA into a host.
They are, yeah.
But when I was reading the section on using traditional viruses, like lentiviruses, systemically in the blood, it sounded incredibly reckless.
Oh, it is highly dangerous. Lentiviruses are notorious for something called random integration.
Random integration.
Yeah, they don't care where they put the payload. They just sort of shove the new DNA randomly into the host's genome wherever it fits.
Wait, hold on. If we're just shooting these genetic instructions into the bloodstream, what stops them from hitting the wrong cells entirely?
Well, nothing. And that is the absolute nightmare scenario the researchers outlined.
Oh, wow.
Imagine you inject a lentivirus carrying the SIRR blueprint into the blood. It's supposed to hit a T cell. But what if it randomly hits a stem cell in the bone marrow?
That sounds bad.
It's terrible. Randomly inserting DNA into a stem cell can disrupt essential genes and trigger transformational mutations.
Meaning it could potentially cause secondary cancers.
Exactly.
That's bad enough. But the paper mentioned an even scarier risk if the virus accidentally hits the actual cancer we're trying to cure. Like, that makes sense to me. Why would that happen?
Think about it this way. What if the virus accidentally engineers a tumor cell instead of an immune cell? It basically delivers the cancer-hunting radar directly to the cancer itself.
It gives the target its own radar. Wait, what does that even do?
It causes a phenomenon known as antigen-negative relapse.
Okay, what is that?
The tumor cell takes the blueprint and expresses the CRIR on its own surface. But because it's a tumor cell, the CRIR ends up bending and binding to the cancer targets on its own cell membrane.
So it's locking onto itself.
Yes, effectively masking those targets. The tumor basically puts on a biological invisibility cloak.
Wow.
So when the actual engineered T cells come looking for the cancer, they can't see the target anymore. The cancer comes roaring back, and it is completely resistant to the therapy.
That is wild. Okay, so the mandate for these researchers was impossibly strict.
Very.
First, you need a way to deliver the permanent genetic payload only to T cells, completely ignoring every other cell in the blood. And second, once you are inside the T cell, you can't just shove the DNA anywhere. You need to insert it into an exact, specific location in the genome.
It's basically like threading a microscopic needle inside a hurricane.
Yeah.
But this new paper shows they actually did it. To solve the targeting problem, they had to invent an entirely new delivery architecture. They abandoned the whole idea of a single do-it-all virus.
Right, because one virus can't do all that heavy lifting safely.
Exactly. Instead, they split the job into a two-vector system, two completely different biological delivery trucks, basically, working in tandem.
I found this split system so fascinating. Let's look at truck number one. They call it the EDV, or Envelope Delivery Vehicle.
Right, the EDV. Its sole responsibility is to carry the CRISPR-Cas9 machinery, the molecular scissors that will actually cut the DNA.
Okay, but how does it know where to go? Because like you said, if it cuts a stem cell, we have a disaster.
Right.
The researchers engineered the outside of this EDV with a very specific homing beacon, right? An anti-CD3 antibody.
Yes, and that choice of CD3 is brilliant for two distinct reasons. First, CD3 is a receptor found almost exclusively on T cells.
Okay, so it acts like a VIP pass.
Exactly. It guarantees the EDV, ignores the red blood cells, ignores the stem cells, and only docks with the T cells.
And the second reason?
It's all about waking the cell up.
Waking it up.
Yeah, so T cells circulating in your blood are mostly dormant. They're in a resting state. But for a cell to efficiently take up new DNA, repair its genome, and integrate those new instructions, it really needs to be actively metabolizing and dividing.
It has to be doing work.
Right. So when that anti-CD3 antibody on the delivery truck binds to the T cell, it mimics a natural infection signal. It forces the T cell to activate.
Oh, that's incredibly clever.
It is. The EDV essentially unlocks the door, kicks the T cell awake, and unloads the CRISPR scissors.
So truck one rolls up, wakes the cell, and drops off the scissors so they can make the cut. But truck one doesn't carry the blueprint for the new cancer radar. That's the job for truck two.
Correct. Truck 2 is an adeno-associated virus, or AAV. Its only payload is the actual DNA template for the CRR.
The actual instructions.
Yes. But here is where the biology kind of fights back.
Yeah, the sources highlight a massive roadblock here because, our immune systems are literally designed to destroy viruses in the blood.
Exactly.
So if you just inject a standard AAV into a human vein, wouldn't the body's neutralizing antibodies just wipe out the delivery truck before it ever reaches the awakened T cell.
Absolutely. The human body is exceptionally good at clearing standard lab care AAVs, and if the truck is destroyed in transit, the blueprint never arrives.
Right. Here's where it gets really interesting.
Yeah.
So the researchers had to do something extraordinary. They couldn't just use a virus off the shelf. They had to build a new one through a process called directed evolution.
Directed evolution, yes.
The paper goes into some heavy detail here, but basically they built a microscopic obstacle course, right?
That's a great way to picture it, yeah. They started with this massive library of millions of slightly mutated AAVs, and they exposed this entire library to human serum.
Which is the liquid part of the blood that's packed with those destructive neutralizing antibodies.
Exactly. And as you'd expect, most of the viruses were instantly annihilated.
But a tiny fraction survived.
Right. So they took those few survivors, multiplied them, introduced new random mutations, and then ran them through the bloody obstacle course again and again.
Wow.
Over multiple generations, they actively forced the virus to evolve evasive maneuvers. Finally, they isolated a single dominant survivor, a newly evolved variant they named AAV-HT7.
It's literally like breeding the ultimate stealth bomber.
It really is.
And the data shows this new variant, HT7, doesn't just evade the immune system. It actually evolved a preference for binding to a receptor called CD7, which is conveniently highly expressed on T cells.
So now the orchestration is just perfect. The EDV finds the T cell, wakes it up, and drops off the scissors. Then the stealth AAV slips past the blood's defenses, finds that exact same awakened T cell, and drops off the blueprint.
Okay, before we talk about where the scissors cut, I really want to pull in one of our supplementary sources.
Yeah.
Because if you are going to all the trouble of engineering stealth delivery trucks to drop off a blueprint inside the body, you better make sure that blueprint is flawless.
Sure.
You can't exactly issue a recall once it's in the patient's genome.
No, you definitely cannot.
And this is where codon optimization algorithms come in.
Right, it's a crucial layer of polish. See, biology has redundancy built into it. When you look at the genetic code, there are often multiple different sequences of DNA-like, different synonyms, that will instruct the cell to build the exact same protein.
But not all synonyms are created equal.
Far from it. Some sequences are incredibly clunky for the cell's machinery to read, while others are highly efficient. So codon optimization uses advanced computational models to scan the desired CAR protein and basically rewrite the underlying DNA blueprint using the absolute most efficient synonyms available.
So it's essentially using software to rewrite the instructions so clearly that the T cell can manufacture the cancer radar with zero wasted energy.
Exactly. You reduce the metabolic burden on the cell. So we now have a hyper-optimized blueprint loaded into a stealth virus, arriving at an awakened T cell that has already been preloaded with CRISPR scissors.
Which brings us to the genetic bullseye. We know random integration is a disaster. So where exactly do the CRISPR scissors make the cut to install this perfect blueprint?
They targeted a very specific neighborhood in the DNA called the TRAC locus.
The TRAC locus.
Yeah.
Okay, let's break down why this specific spot is so important.
Sure. So every natural T cell has a receptor it uses to identify normal threats, like, the flu or a bacterial infection. The TRAC locus is the exact genomic location where the instructions for that natural receptor are housed. The researchers programmed the CRISPR scissors to snip the DNA precisely at this location, effectively knocking out the natural receptor. Then, the AAV slips the new CAR blueprint directly into that exact same gap.
Okay, let's work on an analogy here to see if I truly grasp the mechanics of why this matters so much.
Let's do it.
If the T cell is a CAR,
Okay.
the old lentivirus method of random integration is like taking a massive, powerful new engine and just tossing it randomly into the back seat or the trunk.
Right. And more importantly, with random integration, you usually have to include a synthetic on switch, what we call a constitutive promoter.
Meaning the engine is always running. You toss it in the back seat and just jam a brick on the accelerator. It's running at the red line 24/7, whether it needs to or not.
Exactly. And what happens to a car engine that never stops redlining?
It burns out. Which brings us right back to the exhaustion problem of the ex vivo boot camp. But the Trachy's locus method is completely different. It's like using CRISPR to carefully pop the hood, unbolt the factory engine, and install the new car engine exactly onto the original mounts.
Yes, And here's the genius part of the Trekkie locus. You don't use a synthetic on switch. You hook the new engine directly up to the car's original factory ignition and gas pedal.
The natural promoter.
Exactly. Because the new cancer radar is controlled by the cell's natural internal logic, it behaves like a normal immune response.
Oh, wow.
Yeah, when the T cell encounters cancer, it revs up and attacks. But when the cancer is cleared, it idles. It rests.
That makes so much sense.
Right. This intrinsic regulation prevents the cells from burning out. It maintains them in this youthful, memory, or stem-like state, turning them into a persistent long-term surveillance force circulating in the patient's blood.
It is remarkably elegant. I mean, you've solved the delivery, you've solved the precision, and you solved the burnout.
Yeah.
But elegance on a whiteboard doesn't cure patients. I want to look at the actual data. Like, does this work in a living, breathing system?
It does. The researchers ran really rigorous in vivo tests using humanized mouse models.
Humanized mice.
Yeah, these are mice that have been specially engineered to possess a human immune system, which allows scientists to test the therapy on human cells, but in a living organism.
Okay, got it. And they tested it on a really aggressive leukemia first, right? BAL.
Yes. They infused the mice with an aggressive blood cancer, let it take hold, and then gave them a single injection of the two-vector system.
Just one shot into the bloodstream.
One single shot, the EDV and the evolved AAV. And the data was just phenomenal. That single injection successfully reprogrammed enough T cells inside the mice to completely eradicate the leukemia.
That is incredible. The charts in the paper show it completely outperformed the older random lentiviral methods, too. Like the cells expanded naturally, they didn't exhaust, and they cleared the tumors faster.
It did.
But as incredible as that is, blood cancer is generally where CAR T therapies excel, right? Because the immune cells and the cancer cells are swimming in the same liquid environment. They literally just bump into each other.
It's true. Blood cancers are sort of the low-hanging fruit for this technology. The real proving ground is solid tumors.
Exactly. Solid tumors are a completely different beast. They build these physical, dense walls of tissue. They create toxic microenvironments that secrete acids and signals that literally put attacking T cells to sleep.
They're hostile.
Yeah, so I frankly didn't think an in vivo approach would be potent enough to breach a solid fortress like that.
Well, the researchers had the exact same question. So they reprogrammed the delivery trucks with a new blueprint. This time, they targeted a solid tumor. Specifically, they went after a sarcoma using a B7H3 CR.
And wait, they also targeted multiple myeloma too, right? With a BCMA CAR.
Yes, they did. They proved they could swap the blueprint for multiple myeloma, and then they pushed it even further into the solid tumor space with the sarcoma.
And the results for the solid tumor.
Complete responses.
What?
Yeah. The inside-the-body engineered T cells successfully infiltrated those solid tumors and dismantled them. This really proves the platform is truly plug and play. You keep the delivery trucks the same, you just swap out the blueprint depending on the cancer you want to destroy.
That is a massive victory. But as I look at the future of this platform, and so what does this all mean for the future, I have to wonder how we push it even further.
Right.
Because we know solid tumors are incredibly hostile.
Yeah.
Even if we use the TRAC locus to prevent burnout, what happens if the tumor's microenvironment is just, too suppressive? How do we give these newly minted cells an overwhelming advantage?
And that is exactly where our final source comes into play. If we want to supercharge this in vivo system, we need to look at agents like Anktiva.
Okay, our notes describe Anktiva as an IL-15 superagonist. Walk me through the mechanics of that. Like, what is IL-15 and why does making a super version of it help our engineered T cells?
So interleukin-15, or IL-15, is a naturally occurring cytokine in the body. You can kind of think of it as a broadcast signal that tells specific immune cells to survive, proliferate, and become highly active memory cells.
So it's a natural multiplier.
Yes, but natural IL-15 doesn't stick around very long. It degrades really quickly. Anktiva, on the other hand, is a brilliantly engineered synthetic version of this molecule. It is designed to be incredibly stable, highly potent, and exceptionally long-lasting in the body.
Okay, let me see if I can connect these dots.
Sure.
We use the two-vector injection to perfectly program pristine, youthful CAR T cells directly in the patient's bloodstream. They're sitting at the TRC locus, just ready to go. But instead of just hoping they win the battle against a tough solid tumor, we follow up the treatment by injecting Anktiva.
Exactly. If the AAV is the blueprint and the TRC locus is the perfectly tuned engine mount, Anktiva is high-grade rocket fuel.
Man, I love that analogy.
It fits perfectly. When you introduce Anktiva, it selectively binds to those freshly minted in vivo CRT cells. It floods them with sustained survival and proliferation signals.
So it forces them into overdrive.
It forces them to multiply aggressively and dramatically lowers their threshold for attacking.
Right.
It essentially supercharges them, giving them the sustained metabolic energy required to just smash through the physical walls and toxic defenses of a stubborn, solid tumor.
It's a compounding effect. You make a better cell, and then you give it a better environment to operate in.
Exactly.
Think about what this means for someone, going into the clinic a decade from now. This entire stack of research represents a profound paradigm shift.
It really does.
We are moving away from bespoke, artisan cellular manufacturing that costs millions of dollars and takes weeks of agonizing waiting.
We are replacing the stainless steel bioreactors with the patient's own circulatory system.
It means we are looking at a future of off-the-shelf, democratized therapies. Imagine a targeted injection that can just sit in a vial in a local hospital pharmacy.
Right.
You get diagnosed, you walk down the hall, you get shot, and your body handles the incredibly complex manufacturing process entirely on its own.
Yeah.
It's faster, it's vastly cheaper, and because of the precise TRC locus targeting, it is significantly safer for you and your loved ones.
It is a phenomenal leap forward in oncology. But to fully grasp the weight of what these researchers have accomplished, we really need to look beyond cancer entirely.
Wait, what do you mean? Beyond
Right.
can?
The platform they've built isn't just a cancer therapy. It is a generalized systemic delivery mechanism for precise genetic engineering of the human immune system.
Oh, wow.
So I want you to consider this as we wrap up today. If we now possess the proven technology to deliver complex genetic software updates directly into specific immune cells while they are actively circulating in your blood, why stop at teaching them to attack cancer?
Because you could theoretically teach them to do anything.
Exactly. What if we could design an injection that programs your T cells to hunt down a dormant, hiding virus like HIV?
That would change the world.
Or conversely, what if we wrote a blueprint that teaches your immune system to stand down? What if a single shot could instruct rogue immune cells to stop attacking your own tissue, potentially curing autoimmune diseases like multiple sclerosis or lupus?
Well, that is just staggering to think about.
It is. If your body is the ultimate biological manufacturing plant and we finally have the instruction manual, what will we program it to build next?